So it's a great pleasure to have Andy Dobson from Princeton University here. Andy will be talking about emerging diseases and causes, mechanisms, and prospects. Andy is one of the major figures in terms of wildlife disease and conservation biology. And he has been working with diseases for most of his academic life and in different aspects. Most of them leaning towards a kind of a more quantitative understanding of disease dynamics and spillovers. And he is one of the persons that has been strongly making advances in neglected diseases and emerging diseases and neglected emergent diseases. So all the combinations. So it's a great pleasure to have him here because, I mean, COVID is one of the major examples we have of a spillover or something that is emerging and can be catastrophic, as we already know. So a pleasure to have Andy, and the screen is yours.
Okay, well, thank you very much for inviting me. I really wish I was there rather than here, but it's still nice to give the lecture from here, and hopefully I can come down sometime in the next 2 years or so and give a different lecture, or even a lecture on how we managed to get rid of this. What I'm going to do is talk a little bit about what's been happening as COVID has emerged this year, but interspersing that with other things that we've been working on, looking at other emerging diseases and trying to develop some theory for looking at that. And what theory do we have available for looking at this type of problems? And does it tell us anything about what we should do? I'm going to be informal, so if you want to stop me and ask questions at any time, I'm happy to do that. Similarly, if I'm still talking in about 5 minutes time, give me a heads up and I'll stop talking, or I'll find a way to get through the last couple of slides and wind it up. Okay, this is a section where we think it all started. This is the Wuhan food market in Wuhan, China. The initial cases that were actually reported were reported from here. Retrospectively, we think there may have been cases a little earlier than that. But literally the first indication that I had was this email that comes through. If you're a nerd like me, you get these ProMed emails that come through pretty much every day saying there's some weird disease outbreak. So this one was dated the 30th of December last year, 2019. And it came from ProMed to say, this is strange. We're seeing lots of people with a new type, unknown cause of pneumonia around this food market in Wuhan. And I talked to my friend Peter Daszak and a couple other people, and we said, well, this is a bit weird. We should keep our eye on this, but it might just be another false alarm. We had a couple of false alarm, and the Chinese were very quickly to close down Wuhan, even though they were going into the Chinese New Year when lots of people in China are moving around to go and visit their relatives. Very quickly, Hopkins School of Public Health began to put out data every day pretty much, in fact twice a day, of the number of cases. So as we go, this is from that 10th of January, so that's 12 days after that initial email. There's a small number of cases that are giving rise to an increasing number of deaths. So by the end of January, we could see we had a pretty significant problem. We had over 5,000 cases in China, most of which, 90% of which were in Wuhan, and we had about 150 deaths. So this is about 4 months ago. It was also possible to see as we went from January into February that these cases were spreading throughout China. And in fact, by the time we got to the middle of February, they were beginning to crop up. So essentially, the cat was out of the bag, and we wanted to know, what can we find out and what can we do about this? So essentially, as I say, by the 2nd of March, there are 80,000 cases in China, about 3,000 deaths, and it was beginning to spread globally. So we got it in Iran, we got it in Italy, Australia, and it was beginning to be detected in the US and quite quickly other countries around the world. So if you like, this is going to be the poster child. It spreads very quickly before we do anything about it. Now, it's not the only disease of this type. And in fact, Marble House has been putting together data of just the number of new viral pathogens recorded from humans over the last 120 years. And what you can see here, this is the cumulative number of new virus species that have not been in humans before, have not recorded from humans before. It's been initially increasing at a linear rate, and then from about the late 1950s, it starts just increasing linearly. These earlier changes are because up until about 1890, we didn't know what a virus was. We had no concept of what it was. We knew there was something that could pass through filters that could cause disease. We just developed Cox populus, defined what was a transmissible pathogen, that we didn't have any ability to look or visualize these things. As we went through the 1920s, we began to develop microscopes and things. We could then start identifying things. The technologies got shinier and smarter, but essentially we're detecting things at the rate of about 2 new viral pathogens a year. And you can add to that map some of your favorites. We had HIV, which appeared just as I was a graduate student, probably Pablo was at the same time. It caused a global pandemic with many similarities to what we're seeing with COVID now. We had SARS when we were all messing around doing the global millennium ecosystem assessment, which again disrupted that in around 2000. Several meetings were canceled that stopped us doing that. We've now got COVID-19. 3 years ago, we had Zika virus, even though we'd known about Zika virus since the early 1960s. It was recorded in Uganda then. It just took a long time to move around the world and arrive in a place where it could become, like, cause an epidemic. And so there may be other ones that have come across, and it just in the background before the right conditions come for them to establish. And indeed, if we look at HIV, it seems likely that it initially moved from chimpanzees into humans way back in the 1930s and could just keep going in a series of sort of meta populations that eventually jumped across. So plainly, to understand the dynamics of these diseases, we need some sort of mathematical frameworks. We have the tried and tested SIR models, but we're going to need to have a bit more detail to that structure to understand it. Now, of course, while COVID is emerging in the US, one of the things that slows down our ability to do anything was sort of denial. We had people saying it's just going to be like flu, so everybody should go back to work. And so we were very slow getting going. And the differences now between different countries very much reflects the speed with which they started to do something. Indeed, at the time that we were being told it's a hoax, if you looked at seasonal mortalities in Spain, so this is weeks of the year, this is all the other years of the century, the first 19 years of the century, what's the variation in mortality rates versus this year? By the time you're going into March and April, coronavirus is totally dominating the weekly mortality rate. And Spain had quite a rigorous policy of closing down really quite quickly, even though they'd seen earlier in previous years that elevated this mortality rate, bigger even than we've seen with COVID. By realizing it was a global problem, they closed down and quite quickly were able to shut off this epidemic. New York, in contrast, this is just the weekly increases in cases in New York. It's plainly endemic in the city, and it's also going up faster areas less wealthy people live. And as with the neglected tropical diseases, almost any disease can be designed as a disease of poverty. If you have less financial resources, particularly in the US, where you have to pay for healthcare, you're hit much harder by a disease than people who are wealthier and have schemes in place to making it easy to isolate themselves, but also to go to healthcare and get treated. Now, different groups of emerging diseases, pretty much for the last 20 years or so. About 12 years ago, a group of myself, Peter Hudson, and Brian Grenfell asked a bunch of bright postdocs to do all the work, digging out dynamics about pathogens that are moving from domestic livestock or wild animals into humans. So could we get some basic dynamics at this interface as novel pathogens move into human populations? And you can go and the postdocs did a fantastic work. Essentially did lots of editing without much intellectual input, but essentially what the type of patterns you see in the middle here is the sort of classic epidemic. Disease moves into in red, moves into a population of susceptibles in black. It generates an epidemic which reduces the population of susceptibles. If people recover, and eventually through time, you hope the population becomes dominated by immunology-resistant individuals, which slows down and hopefully eradicates the disease. Or it allows it to persist at a very low level. I'd like to speed this up if you had a vaccine, but by definition, if you have a novel disease, you haven't got a vaccine. What you also hope is if you have a treatment, you can treat people as they get sick or prevent them from getting sick. If you treat them when they are sick, they will become immune, you hope, cause this sort of dilution effect, or herd immunity, as it's called, which slows the dynamics where the average rate of transmission is one infected individual that's only able to infect one other individual. Early on in the epidemic, that's not the case. We have this number R, which is the number of people that the first person is able to infect, and that R0 number diminishes through time. But our worry is, what is the magnitude of this? Because it determines what we get expressed. Now, there are various stages. Your ability to estimate that number is crucial, but it's going to be a really fuzzy estimate early on because you haven't got that much data. So as the epidemic speeds, you get better data that allows you to get a better estimate of its transmissibility, but at the same time, you're either trying to control it by killing vectors or getting people to social distance. So you would expect it to be going down. So your ability to estimate it gets better as you get more data, but it is changing all the time. So what you want to know is how does this respond to when control begins, and what are the future trends going to be, and your ability to predict what it's going to do next is very much a function of the quality of the data and the structure of the models you have about how those allow you to look at what it's going to do in the future and respond to different control strategies. Some things may be dependent on climate, particularly if they're vector-borne diseases. Like in the case of COVID, we don't see a strong climate signature. But of course, if everybody is susceptible, hugely masked, we don't really see the effect of climate in many of these until they become endemic. That again is confounding our ability to do things with it. What happens is that you get an epidemic of papers on it. So filtering through these to try and find out what's going on. This is SARS, the original SARS that came across, as I said, towards the same beginning of this century. Boom, the number of research papers is going up massively. The number of modeling papers is also going up rapidly, but about 10% of those papers. Borrelia, which has been around for longer, has hundreds of papers, but relatively few modeling papers. Now was sort of acknowledged because it was out there for a long time, and then suddenly it creates an epidemic when it moved into the US in about 1990. A handful of modeling papers, but tons and tons of information. Because it's a vector-borne disease that generates immunity, it more or less takes care of itself and begins to sort of decrease to levels where it's not a huge problem in the human population. So let's look at one of these in more detail. Before that, let's look at the type of things that cause these pathogens to emerge. And if we look at them, SARS, MERS, COVID, HIV, there seem to be two major broad classes. One is the wildlife trade, which is a sort of trade in species that are used for medicine, for food, for the fashion industry, in the case of fur coats, for a variety of different luxury goods. This is the world's fourth or fifth biggest industry. It's regulated by an organization called CITES. The budget for CITES is about $6 million. This year it's $1.4 million because none of the people who've signed up for CITES have actually given their money yet. So we actually have a pathetically small amount of money for monitoring. It's so easy for them to jump across. If we look at the amount of trade, these are just data. Every country, 183 countries have signed up to CITES, which means if you divide $6 million by 183 and different countries paying different amounts, some countries are paying like $1,000 a year to CITES. But if you go to Singapore, which is sort of representative country with relatively strong governance and well-connected trade, you can see that since CITES started in the mid-1970s, trade has been steadily increasing. And in particular, as China's economy took off this century, more and more stuff is coming into trade. I've colored this by different groups, bird species, mammal species, but we can divide it, just do this way, by the different groups. This is birds, mainly going into the pet trade. It's not numbers of birds, it's numbers of species of birds. So at any year going through Singapore, 100 to 180 different species of birds all going into the pet trade. Mammals, to food rather than pets, there are 60 to 80 species with the numbers consistently going up. Plants, so it's mainly sort of house plants and decorative plants. Again, it's a large number of species. Reptiles, this is again 30 to 40 species. Corals and marine invertebrates, again, many species. Fish steadily increasing. So you got lots of species going through. An interesting thing is if you just compare total imports of everything into the US with into China, you see two very different patterns. The US signed up to CITES early on. As CITES gets going, but it's essentially constant. The US imports 3,000 different endangered species or species a year. And this includes everything from elephant tusks to things for the pet trade to weird things for people to eat. China has been steadily increasing trade as its economy has strengthened, but it's still an order of magnitude less than the United States. There are distortions and biases in this data because depending on how things are reported, you'll get better quality data. So this is legally recorded. There may be factor 10 to a factor 100 differences in these data, depending on how well this is policed. But as I said, with this rather pathetic budget of $6 million, we desperately need to know more about this. That COVID came out of something in the wildlife trade. And as I said, we also know that that's the same for SARS and also for several of the other emerging diseases. Now, the other source of emerging diseases, and this map doesn't work, we know that habitat conversion is a major driver of global change. It's a major contributor to climate change. It also is the source of many emerging diseases. If you look where Ebola has emerged in Central Africa, it consistently, it's emerged about 10 different times, emerges along the edges of the forest, where what natural forest is being converted into agricultural land. It's usually the people doing this conversion are the first people to be infected, or their children. Usually it dies out because you have relatively small populations in these logging camps or in these little communities. Tiers of conversion, but occasionally, as these areas get better connected by roads, and we've seen this twice in the last decade with Ebola, with a huge epidemic in West Africa and an ongoing epidemic still in Central Africa of Ebola. Primarily because people are eating things whenever they find them or can capture them, where they're converting forests into new habitats. So again, all of the sites this barrier. Now, we can make models of climate change. Curiously, they have the same structure to the SIR models we would look at for looking at diseases. So the disease models have susceptible hosts, which would be pristine habitat, agricultural land, which we can think of as land infected with humans, and degraded land, which is land that's no longer suitable for agriculture. So those would be the same as immunized hosts. Okay, so that has to come down there. Okay. We can also have land that's made resistant because it's converted into urban areas. Now, if we look at different parts of the world, the Pacific coast of the US, you could do this for all the different areas of the US. We see this dynamic that's like a very slow form. Pristine land, forest, is converted to agricultural land, which becomes abandoned and becomes degraded land. It's called recovering or degraded depending on how it's managed. So you could write that down as an SIR type model. You can add humans and do the dynamics of that. They're very similar to the dynamics of an epidemic. This is the infected hosts. This is the susceptible hosts which have declined. This is the recovered hosts, or the agricultural land that's been worn out and become degraded, eventually restored to the forest, which is similar to people losing immunity. And the relative proportion of land is driven by the relative length of time you last in each stage of infection. So the longer you last as agricultural land, the more agricultural land you have, the less forest you have left. So there are interactions there that very much match those we see as the classic disease. Now, once you have a framework like that, what we were interested in doing with a group of us initially at NCEAS and then at CISINC in Annapolis was to say, could we look at disease transmission types across this boundary between forested land and human modified landscapes? Types of pathogen that might be transmitted from monkeys to humans, say in the forest, or from monkeys coming out the forest into humans, or from could even go backwards from humans, can take COVID, or they could take influenza or measles back and infect disease, or wild animals might affect domestic animals, or vice versa. We would also have vector-borne diseases, and we might have trains of transmission going on out between the two. So there are different ways of looking at how these things are nested, and what are the changes in environment, the changes in animal movement, range expansions that lead pathogens A different group of us tried to look at how this might work for different things, such as Ebola virus, E. coli, for Borrelia, for Sinombre virus, which is essentially hantavirus. I classify those. But a different set of us said, well, let's go off and look specifically at some of these pathogens in the field. So this is a place I went to about 20 years ago. This is the index farm, but an emerging disease called Nipah virus. And Nipah virus actually scares me more than COVID, partly because it's very similar to measles, which means it's more transmissible. It has even nastier or slightly nastier symptoms. This is the farm with the pig farm on the Malaysian peninsula. And it's very hard to get into this farm because it's protected by these sites. We accept no visitors in view of maybe swine elsewhere. This is the only protection to stop you going into that farm. When you go into it, it was all destroyed, killed, and they tried to initially knock down the farm until they lost interest. This is actually the physician who went in and identified Nipah virus as a problem. We went there with him, and he was explaining that what seemed to happen is that pigs were a major industry in Malaysia, and so were mangoes. Mango trees, sorry, not mangoes, mango trees in sort of natural little forests of mango trees because the mangoes supplied shade, because you don't want your pigs overheating. The mangoes also drop down into the farm. Pigs get the extra meal of mangoes. I really like mangoes, and so do the pigs. The problem is somebody likes mangoes. I'll get back to that. The other things that really like mangoes are bats. And so if the bat's been trying to eat a mango, it tends to spit out the seed. So a seed covered in mango saliva gets spitted into the pig farm. The pig eats the saliva covered mango, and that can potentially cause transmission of a pathogen from the bat to the pigs. Now, this is something you rarely see. There's two people in this slide. The guy on the right is Peter Daszak. You may have heard about Peter. If you've been in the US, you will have heard about Peter because he's been working on emerging diseases for the last 20 years. His arm was cut off by NIH 2 weeks ago because he'd been working with the people in the lab in Wuhan in China. The guy on his left is even more unusual. This is the patient in the Nipah virus epidemic. This is the guy who owned the pig farm where the pigs got infected. He got infected by handling his sick pigs and got really ill. He infected his family, and infected some other people, and that's why the physician went in to treat them. Luckily, he survived, but he has pretty pronounced neurological damage. It's one of the few examples we have of actually a patient zero. So let's go into this in more detail. These are the bats, and the bat is eating mango. Because bats fly, they don't want to carry a huge, heavy weighted mango seed in their stomach, so they tend to suck all the fluids out of the mango and spit it out. It gets spat into whatever's beneath, which in this case is a pig farm. So it's a great way of transmitting a viral pathogen from a bat to a domestic host species. So we started doing surveys of Nipah virus around the Malaysian peninsula, and these circles just indicate the bats that have been infected, because this is serology. About 70 to 80% of the bats throughout Malaysia have been infected sometime with Nipah virus. Those that are actively infected is always much less than that. It's always of the order of 1 or 2% because it's a classic SIR type pathogen. You get infected and then you become resistant to measles or chickenpox. But it's plainly at high prevalence around the island. It's incredibly pleiotropic. There are no genetic differences between bats from anywhere right down to Sumatra and across to Borneo. They're flying around and constantly switching between colonies, so they can easily move these virus pathogens around. And in fact, the more we look at it, there is something unusual about bats. Say, COVID seems to have come from a bat. Nipah virus and Hendra virus come from bats. Ebola comes from bats. So one of the things we need to know is, why do we get so many weird pathogens from bats? Surveys that Kevin Olival and Peter Daszak and some of their colleagues have been doing, we can show that bats and other small mammals have quite rich viral flora of pathogens, as do many other groups, ungulates and primates. Bats somehow have a subtly different physiology, which means that the pathogens that are in them are not particularly unpleasant to bats, but really unpleasant to other mammals when they get into them. Bats have a subtly different pathology because their physiology is very different. Having to fly gives you a different physiology than when you're a non-flying mammal. And the simplest component of that is, if you're a bat and you fly, you have hollow bones. If you're a non-flying animal, you have bones of bone marrow, and bone marrow is where you produce the B cells that drive your immune system. So subtly, bats must work in different ways. And with a student, Cara Brook, we've been looking at this and trying to make within host models of the immune system of bats to compare with within host models of the immune system of, say, cats and dogs and mice, to see how are the dynamics of viral pathogens different in bats than they are from these in these bats. Now, if we go back to Malaysia, bats infect the pigs. If you look at the distribution of pigs throughout the Malaysia Peninsula in about 2004, there were about 6 million pigs in Malaysia. They're exported to China, they're exported to Singapore. It was the major agricultural industry. This is the density of farms throughout Malaysia. On the right-hand side is the density of mangoes, which is a pretty close match, because, as I say, they use the mango farms, and that allows transmission of virus into bats into pigs. It then gets curious. This keeps locking on me. Hold on. Yeah. So if you just look at the expansion of agriculture, so you've got the deforestation leading to, they leave the mangoes, but they start pig farms. Pig production goes up. It's the standing pig population in millions, up to about 3 million. This is mango production in tons, because it's an associated crop, but it also feeds the pigs. We then get this huge outbreak, and more than half the pigs in Malaysia are destroyed to prevent the epidemic spreading. Now, the epidemic is interesting, and this we think is characteristic of many of these emerging diseases. When they destroy farms is about March 1999. The guy that I showed you the picture of was actually infected in January 1997, way back 2 years earlier. There'd been little outbreaks, and then for some reason we suddenly switch and get a much, much bigger outbreak. Now, what's been going on here? Well, Juliet Pulliam, who came to work with me, gathered all the data for all the pig farms and the pig farms. What you can see is that there are 2 different periods. What tends to happen is, if you have a population of susceptible pigs, as I showed you in that earlier slide, you introduce an infected mango into pig, you get an outbreak which kills a lot of pigs, or they destroy a lot of pigs, you generate resistant and recovered pigs, and then you have new pigs that are born to susceptible models, and the epidemic's gone away. Instead, what happens is you then get a second introduction of the pathogen. And because you've got a population that's partially resistant, because its mother was infected or because they survived the previous epidemic, when you get a second epidemic, it's not as big as the first epidemic, but that means it lasts much, much longer. It slips from becoming epidemic to becoming endemic. And that, of course, is our worry of what's going to happen with COVID. We've seen a massive epidemics every time it's arrived in a new country. We now appear to be in this period where in many places it's declining, some it's still going up, but people are beginning to think it's going to disappear. What's going to happen is it will be reintroduced from a different country as travel restrictions are lifted. We won't get as big an epidemic as we've had before, but that will likely allow it to become endemic. And indeed, what Julia did in a research paper disappeared completely, almost without trace. It was in PLOS Biology. She worked out the proportion of time, the rate at which immunity is lost against R naught established on its first outbreak, but the second time it appears, it has a much better chance of establishing if you have seen immunity. So if you look at different diseases, measles, rubella, pertussis, it seems to be characteristic. Once you have this immunity, you get a dramatic first epidemic and then a much more prolonged second epidemic, which switches from being epidemic to becoming endemic and with you all the time. And we suspect this is what's happening with COVID. Now, at the same time, and this was all on the same grant, also working with us was a student called Raina Plowright, who went to Australia, partly where she came from Australia and went to work. There's a sister pathogen to Nipah virus, and that's called Hendra virus. And it's present in the fruit bats, the pteropods, radiated as a big radiation of bats in Southeast Asia. The ones in Australia are distinct from the ones in the Malaysian peninsula. The best ones are in New Guinea because they're all called Dobson's fruit bats. So it's great that there are all these fruit bats named after me. What Raina did was to go and look at in Australia and showed that there were lots of what are called camps, big colonies of bats all along the coast of Queensland. We started making models for these bats and for Hendra virus, and what you could show, if you look at the initial hosts in the population and the size of the epidemic, when the pathogen first appears, you don't have any, a big epidemic, but it dies out. Once you have a proportion of the population, the epidemic isn't as dramatic, but it goes on much, much, much longer. So one of the key things with these diseases is how long they last relative to the proportion of hosts that are immune. And of course, one of the things we hope to do with the vaccine is to vaccinate people to protect them, but the downside of that is by increasing the proportion of hosts immune, you will increase the persistence time of any future outbreaks. So let's look at another part of this, which is, as I said, as we convert natural habitats, essentially what we're doing is taking a forest, convert it into agricultural lands, whoops, let me go back, and creating a different type of habitat matrix. Now, to a simple approximation, if you're worried about transmission from wild animals into humans or human animals, the rate of transmission is a simple function of this perimeter between natural habitat and converted habitat. And this perimeter will increase as you start converting habitat, and will decrease as you run out of natural habitat to convert. So to a rough approximation, the dynamic, the rate of transmission will peak at intermediate levels. And it'll be a function of the number of the hosts in the core habitat and the number of potential hosts in converted habitat. So as part of this working group, at NCEAST and CSynC, we looked at models for this, thinking about two different types of transmission. Density-dependent transmission, which would be like COVID, where the more susceptible hosts are present, the more hosts an infected individual is able to transmit the disease to. That contrasts with something like frequency-dependent transmission, where the proportion of hosts you can transmit to is roughly decreases as hosts become more abundant, because you can't contact them all. So essentially, thinking mechanism of transmission, coughing and spluttering and sneezing and leaving virus-covered fingerprints everywhere, you might have dependent transmission. If you're using a vector, or you can do the Freudian experiment of thinking of a sexually transmitted disease where you've got a handling time, that means transmission is finite and there's a finite number of hosts you can contact within a given time period, then transmission is likely to be frequency dependent. You can also have relative rates of within and between species transmission. As rates of between species transmission to match within host species transmission, then you will get obviously more transmission between species. So as habitat is converted, rates of transmission for density dependent pathogen will peak at intermediate levels but decrease rapidly at either no habitat converted or all habitat converted. Similar things will happen for frequency dependent transmission, but less symmetrical. You can also look at epidemic size, because you can do a simple stochastic version of this. So your probability of getting an outbreak is always at a maximum once you get above about 25% conversion, about 75% conversion. But the size of the epidemic is bimodal. As the proportion of habitat converted gets bigger, you've got more and more hosts, humans or domestic livestock in the converted habitat. So you either get a small epidemic because you're reducing, or you get a massive epidemic because you've got such a huge susceptible population for the pathogen to move into. And again, this is what seems to be happening with COVID. At lower levels of transmission, we get these small epidemics to keep dying out because the proportion of hosts in the habitat, size of the populations in the converted habitat is relatively small. At intermediate levels, we will get quite large and expect to see quite large epidemics that still die out. Once we've converted about 90%, we're beginning to see a diminishing probability, but if we do get an outbreak, it could be huge. Now, the other thing we wanted to look at with this working group is how does biodiversity of all the different species in a habitat affect these things. And this is, again, a very nice paper he did with Peter Daszak, I think it's in Nature. What's the diversity of potential reservoirs of bats in habitats around the world? Well, this is a huge diversity of bats in the Amazon. That should be a relatively risky area for new pathogens. Carnivores seem to have a higher density in East Africa, as do ungulates, so we might expect more of those types of pathogens to come there. Primates, again, it could be somewhere from Africa, it could be South America, or it could again be the Malaysian. Small mammals, again, it's worrying from South America, from Africa, across the Russian steppes and in Western North America. These different groups have different diversities. What we can do is to actually have more people out there trapping animals, taking tissue samples and seeing what's out there and trying to create an on the web encyclopedia of what's out there, because knowing which different viruses are out there, identify where it might have come from when we see a new one, it will also help massively with the development of accurate tests for these things once we have to start testing people. So again, this group of people at NCEAST and CSynC put together a paper looking at the community. As landscapes get fragmented, going from large areas down to small areas, the number of the species diversity in these patches decreases, and so does the average body size of species in these patches. So in large patches of habitat, you have a large diversity of host species of a range of body sizes. In small habitat patches, you tend just to have species with small body sizes. You can take your standard SI or SIR model, rescale everything by body size, because things like birth rates, death rates, population densities all scale by body size. Once you put them into that framework, you can transmission of different pathogens that will give you R naughts of different values. Will also scale with pathogens that are density dependent, having to have higher transmission rates as body mass gets bigger because there are lower population densities. The parasites will have lower rates of transmission for hosts of bigger body sizes because they will have more contacts per lifetime with other host species. So you can rescale body size. You can show that the relative abundance would change more or less as you see changes. It'll give you a species area curve that corresponds to the sort of species area curve we see for mammals. So essentially you can recapture this. And then ask the question, what is the relative risk of different diseases being covered by the host community or being able to break away from that host community? And roughly what you see is if frequency-dependent transmission, then the risk of a pathogen outbreak is much lower in an intact habitat. In contrast, if you have density-dependent transmission, as you start fragmenting the habitat, you increase the risk of transmission in wild hosts with natural hosts. So I'll skip over that. Now, returning to COVID, sort of briefly, we're beginning to see this is up until the end of March when everybody's now in lockdown, looking at data for different COVID. This, I think, is really interesting way of plotting out the data for COVID, because it's essentially saying how many cases have you had against time, but for starting at when did you have the 100th case, and what's the trajectory of different countries. Here, contour lines of cases doubling every day, every 2 days, every 3 days, every week, and show countries are doing different things. Essentially, you're seeing the response to either a rigorously applied lockdown, or a test, testing people. But plainly, some countries are not doing a particularly good job. Essentially, the US, it's doubling every 2 days by the time we get to the end of March. Spain, it's doubling every 2.5 days. The UK is pretty consistent on 3 days. Japan, Singapore, and Hong Kong got going quickly and began to close the thing down. Now, part of this was isolating people. There's lots of talk about testing people, but testing people is quite a tricky thing, as we're beginning to see in the UK at the moment. Initially, we have tests. We stuff something up somebody's nose or up their mouth to try and get a sample. That's trying to see if you've got virus in your airways, whether you're actively infected. Just because you've got a set test doesn't mean it's consistent. Prior to showing symptoms, there's still a relatively low probability of that test being positive, which means lots of people are able to transmit at a relatively low level, not knowing they're infected. Once they're infected, you begin to have a better chance of detecting the virus using this test in their airways. And again, your ability to detect the virus decreases through time. Second type of test is one that looks essentially saying, has your immune system kicked in to control the volume of virus in the blood and provided with you with antibodies which will hopefully protect you. In many cases, the virus virus was. These are actually people who are severely infected. It's the antibodies that are killing them more than the virus. But at least you can see, are they now protected? How long will this protection last for? And could we induce it with a vaccine? So there's a lot of needs to be tested, but the way you're tested with a blood test versus a swab test will consistently change through time. Easter was very different this year. Self-isolation was crucial for everybody at Easter. On a slightly different tangent, but bring us back to this thing about testing, about COVID. Now that might seem a strange way to go, but one thing we've been working on for the last 4 years now is an emerging disease of these birds. Birds on the feeders that are literally 5 or 6 feet away from me. I have them surrounding my office here. These are house finches, passerine birds that mainly live in the Sonoran Desert of California, and Arizona, New Mexico, and go right down into Mexico itself. But they produce in the eastern US. So in the eastern US, where I live, we tend to think of them as birds that come into our fields. Their natural habitat is actually saguaro cactuses. Now they're interesting because they're an invasive species in the eastern US. They're a native species in the western US. Some nutter introduced them into Long Island. And these are data from the Cornell time of the abundance of house finches, just by people going out to houses. You can see they begin to increase in abundance. In the United States, they're ticking over at roughly constant abundance in the West. Increasing as an invasive species, they begin to spread, spread across to join their native part of their range. Their abundance begins to decrease. In the east, even though they're still spreading, their abundance, as I'll show you in a minute, has decreased by about 50 to 60%. Now this happened because the disease escaped from poultry farms and house finches. So I showed you a relatively healthy house finch in that first slide. This is what a house finch infected by this bacterial infection might look like. Its eyes are really badly screwed up. Now usually it's only infected in one eye rather than the other eye, but that being infected certainly reduces its ability to find food, and it also increases its ability to be attacked by predators. So we know that this disease came across in October 1993 because we first started seeing infected house finches like this just outside Baltimore in October 1993. The nice thing about working on birds is that there are thousands, hundreds of thousands of people out in the US looking at birds, and just like me looking at birds. Something weird happens, we pick it up quite quickly and we can monitor it. These are all the people looking at birds visiting their fields. This pathogen just jumps across and starts reducing the abundance. So it's an interesting model that has several important similarities with COVID. The chief difference is nobody cares about this pathogen. There's a bunch of birders who care. There's not enough of them to raise the money to try and eradicate the pathogen. So it's one of the very few examples of an emerging pathogen where we can just let the system run free, see what the pathogen does to the host population, monitor its whether its virulence changes or not. And of course, that's the next thing we need to worry about with this COVID pathogen. Is its virulence going to change? So it's also interesting that you can see if a bird is sick because we start seeing these lesions around its eyes. And because it's birds and we have the wonderful Cornell lab of ornithology, we can set up a membership forms They now do this on computers, but this is actually done before we really had use of computers. Think back to those days, where people would record every day of the month how many sick birds they'd see coming into feeders. So this isn't the distribution. This is the distribution of people actually recording this data across the US. 20,000 people recording data so they can record the epidemic live. And what we see is this epidemic out from its origin down here, just outside Baltimore. It spreads as a wave across the US. 1994, a year after it first appeared, 3 years after it spread, it was just behind the expanding wave front of the population. Now you can see it slows down in mountain areas of Appalachia. We're lucky in that working on the birds are pests. We don't collect birds to work on. In upstate New York, where they have this thing that might just be usable as vinegar, but they call it wine in upstate New York. In California, it can work in both places, but essentially they will give us wine to go and remove birds from their vineyards, and then we can bring them back and do experiments on them. Now we can use the citizen science data collected by people looking at their feeders. This is the house finch population in Virginia, Maryland, and Delaware. This is central Pennsylvania, eastern New York. Their abundance increases of Long Island, and then the disease arrives and knocks down their abundance to 60% less than it was, 40% less than it was. So the disease definitely has an impact on the population. We lost of the order of 500 million disease establishing, and the numbers have never recovered. If you look at the way it established, the details are really quite interesting. This is the decline. It's established down. This is in slightly more detail. What these red dots are the size of the social group that the finches are living in. So the disease knocks the abundance down, massively reducing the social size of group that the birds are living in. They decrease from about 20 group sizes of 5, and over the course of the longer epidemic, sizes of about 18, as I say, down to groups of about 5. So all the reduction in population size is really a reduction in the social group that you're living in. A wonderful metaphor for social distancing. The social group, the disease will ultimately reduce itself and become endemic, and this is effectively what's happened with this house finch. All the different sites when we sample them, so this is like site occupancy. The site occupancy goes way down and then slowly comes up but doesn't recover to the level it was before. These are the rates of extinction. Sites are constantly going extinct, but then they're recolonized initially at a higher rate. So we can monitor this in great detail, and you can show that the relative change in abundance. If you look at the percentage of birds, the probability of a bird seeing a sick bird at any one time, notice it's not dramatic. Although lost half the birds in the population, we never see more than 2% of the hosts infected. People in, their abundance comes down, and we end up with of the order of about 1% of the birds infected, causing a net 40 to 60% reduction in host abundance. Now we can do all sorts of interesting experiments on this. We can show consistency. We need this bacteria to actually cause the infection. We do this with birds in the eye with a stick. This is a medical swab, similar to sticking a swab up the nose of somebody with COVID, but the bird is fine 5 minutes later. You can put this on a gel and consistently show. So that's that bird after sampling. We poked it in the eye with a stick, but really it's the pathogen, not by the stick. We know it's a sick bird. We can take that sample and infect another bird with it and do all the cost postulates. Now, why is this relevant? Look at the progression of the disease. So this is a healthy bird. It has eyes not infected. This is one where you're just beginning to detect the infection. This is one with a pronounced infection, and this is one with a really bad infection. Now, if we look through time, this is the I score. Uninfected, 0, up to about 2.5, a really badly infected. This is the ability of a bird with that level of infection to transmit the disease. Now, what you see here is important. We're sort of following trajectories through time. The bird's ability to transmit the disease goes up very rapidly before you even see that their eyes are infected. Once you begin to detect that their eyes are swollen up infected, the immune system's kicked in and the bird is actually becoming less infectious, even though the eye score is pretty bad. But that's because the pathology you're seeing is this interaction between the immune system and the presence of the bacteria. Once the eye score goes down, the bird hasn't been capable of transmitting the pathogen for several weeks now. So there's a disconnect between when the bird is able to transmit the pathogen and when you actually see the pathology. And that is crucial to other types of pathogen. This is a beautiful paper that Christoph Fraser and Roy Anderson did back in 2004 when we first had the SARS. If you look at the relationship between transmissibility of a pathogen, it's infected in the early stages of the epidemic. So an individual can infect 4, 6, 8, 12 hosts, versus the proportion of infections that occur prior to symptoms or by asymptomatic infection. SARS and smallpox can have quite high R naughts, but most of their transmission occurs once you can see symptoms. And that makes them very easy to control. So SARS appeared, as I said, around about 2000, but most people's symptoms didn't appear until they were infectious. Just by isolating, made it easy to control SARS. Smallpox is hailed as the great achievement of human disease we've eradicated by vaccination. But again, you could go along with isolation because the symptoms of smallpox appear when people are infectious, but not a lot before. Contrast, influenza, which we've had for at least 400 or 500 years, you can transmit that for 40% of the time before you actually show symptoms. And HIV, the biggest pandemic ever, 90% of the time that you're infected, you don't know you're infected. And that's what makes it very hard to control. If we look at COVID, this is the one thing we began to realize by the time we get into March, is that COVID is very different from SARS. Lots of the transmission is going on people's symptoms, and that's going to make it hard to control. So if we put COVID on, somewhere between 25% and 40% of transmission is occurring before symptoms, which means it's going to make it quite hard to control. And that's what we're wrestling with at the moment, the fact that we have these tests for it, but the tests aren't particularly good before symptoms appear. It gets much better once people have recovered, but by that time, there's no point isolating them because they've recovered and maybe immune. Now, how does that affect the people? How does, when the symptoms appear, affect how virulence is going to evolve? Now, the nice thing about this house finch system is if you've been able to study it for 30 years, it emerged on the East Coast. We're constantly taking samples and studying the birds in different places so as we can look at a phylogeny of the levels of virulence, how quickly the I scores go up. So if you look where it's been endemic, it consistently evolved to become more virulent. It's much nastier now than when it first emerged. It also, as I showed you earlier, it spread west, following the hosts, but as it spread west, it also did this thing of becoming less virulent. It was just coming to California and feeling less aggressive. But as it spread west, it became less virulent until it became endemic in California, when it again then started to become more virulent. The pathogen is spreading, its initial spread or its later spread as it spreads across the country, it is evolving to become less virulent. Wherever it's endemic, it becomes more virulent. So we can show that it was mainly a single strain in the population that moved across to California. At first we thought that's good, it's evolved less virulent strain. We're hearing stories for COVID of reductions of virulence in Italy, which I think is a wonderful naive thing, because if it's reductions in Italy, it doesn't mean it's going to do the same thing anywhere else, because the Italian population is not connected to any of the rest of the population of COVID now. Nevertheless, what we see is it's been endemic, it's become more virulent. As it spreads, it becomes less virulent. As it's endemic again, it becomes more virulent. Now we can again make diffusion models of this. We're looking at trade-offs between transmission and virulence. Look at that, and what you see is that if you're just thinking of a pathogen that's dispersing versus the one that's become endemic, when you disperse a susceptible population, you're likely to evolve to a lower level of virulence than once you're in an area where you've got a trade-off between movement and virulence, or a trade-off between virulence and transmission. And as I say, we can look at all this. We know it's only in we do experiments trying to tease this apart, looking at the different strains of the pathogen, the different host strains. But essentially, what you can show is that if this pathogen produces immunity, once it gets into a population where herd immunity begins to increase, then it will evolve to become more virulent because of the immunity that it produces and its ability to establish in the population. So the interesting thing about this is it contrasts with the classic coalition story based on myxomatosis. In myxomatosis, there's a trade-off between transmission and virulence, which means if your virulence is too high, your transmission goes down. But this housefinch bacteria, and we think also for COVID, there's no trade-off between transmission, or a much reduced trade-off between transmission and virulence, because transmission occurs before virulence is expressed. Instead, you've got is a trade-off between immunity you're beginning to establish, and if strains can build up higher levels of immunity by being more virulent, then more virulence will be found. That worries me about COVID, that this thought that it's going to evolve to become more benign may be completely back to front, because there's no constraint on virulence, because it can occur before, because transmission occurs before virulence is expressed, then COVID may evolve to become more virulent. So you can do the math on that. The very funky thing is that if you look at how this virulence is evolving, we can do all the genomics with colleague Scott Edwards at Harvard. It turns out that the genes that are expressing virulence are actually CRISPR locus. You know, there's all this excitement about CRISPR because it's used as a tool for gene editing, but its actual role in bacteria, CRISPR is the immune system of bacteria. It's how bacteria identify phage that are trying to kill the bacteria. Now, what's happened with the housefinch, we think, is that once it goes from being chicken, where it's constantly attacked by phage all the time, once it moves to the relatively immunologically benign side of being in the eye of a finch, it's not attacked by a phage because there are no phages in the new host. That's another wonderful example of a pathogen being an invasive species that escaped its natural enemies, the viruses that attack the bacteria. So it doesn't need its close to operate as a viral identification and mechanism anymore. It activates itself as an increased attachment mechanism to make attachment more easy, and also generates a bigger immunological response that prevents other strains getting. It's actually the most bizarre example of CRISPR actually being the natural history of CRISPR actually being looked at in the wild, but what is actually driving the evolutionary process. So anyway, just returning to sort of wound up, we're seeing a whole studies of these viruses. We're not yet at the stage of looking at evolution. I suspect that's we'll start getting data from that in about 18 months, 2 years time. But the assimilation of I think may well turn out to be naive. Once we're throughout the spring, we've seen amazing work. Some of my favorite is this work from Italy that just February the 25th to March the 25th, this pathogen spread right through Italy, causing huge amounts of mortality. But again, we could look at the models we had, looking at a spread and what we need to do to try and control it. Similarly, the genetics work looking at the structure of from China, but spreading around the world so that we can identify where it's come from, but plainly it's now everywhere. The Chinese, we've all seen pictures of them building these hospitals. The Brits also managed to build hospitals very quickly. They managed to turn it around, but I think it's too soon to be optimistic that we've got rid of it, because if it's everywhere, transportation, it'll come in from other places. The US, cartoon appeared once they made Pence in charge of coronavirus. At the moment, I think it's coronavirus 100,000, Pence 0. And we definitely need organization and international organizations to have an international response to this virus. No country by itself is going to be able to deal with it. Wearing masks is still the best thing you can do, and also in the back of our minds, this is a huge existential crisis, but it's pretty just good preparation for the other one that's coming along sometime soon, which is going to be climate change. Okay, so what have I tried to say? We've learned a lot about COVID very rapidly in the last 4 months. I think we've been able to do that because we have a fantastic body of theory and lab work that put out by Roy Anderson and Bob May, but their work on the infectious disease, a fantastic mathematical template for which to look at this disease. Other work that people suggest that prevention and stopping is the best way to deal with it. So I am happy to answer questions. Thank you so much for inviting me to give a talk. I hope everybody can stay safe.
Thank you, Andy. Great talk towards the force on epidemiology and wildlife diseases. So
right,
thank you very much.
I've talked for like an hour and 20 minutes. My apologies.
No, this is great. We have plenty of time.
Oh, good.
So I don't know if anyone wants to ask questions. You just can raise your hand. Just click on the participants, and I'll have a list here, and I will see if anyone has raised their hands. I have a couple of questions for you, Andy. This is a great kind of an interesting talk. I wonder. Always I've been kind of wondering about the endemic state of many diseases. And as you showed, many tend to actually evolve towards that endemicity because of the immune response of the host, and you become less virulent, which might be kind of, they still kill or killing a fraction of the population each time, but it's something that you get used to it. It looks like MERS is going that way. MERS, because it's killing, it's very aggressive and it's killing always. Is I think 2019 was like 800 people died from MERS, which is, I guess, transmitted by camels at some point. So the question is, how many endemic diseases can a population actually resist before going extinct? Because, I mean, we can keep accumulating HIV, MERS, many of the viruses that are still killing us, but eventually they might start interacting somehow and kind of adding effects.
That's a, I mean, a series of very interesting questions. I mean, straight off, these pathogens, any pathogen can become endemic without changing its virulence. You don't have to have any loss of virulence to become endemic. All you've got to do is generate herd immunity. And once you've done that, you can settle down, because the birth rate of the population supplies a constant supply of new hosts for you to infect. I mean, measles has been endemic in humans probably since the Athenian plague of 350 AD. We know there's been no genetic selection in measles since a selective sweep in the 1930s. And it's still a really nasty disease. I mean, people think, oh, I mean, because so few people get measles, we tend to think it's not very, very pathogenic. But one thing we're going to see with COVID is that because people haven't been vaccinating their kids for measles because of isolation, we're going to see some pretty significant epidemics of measles, and they're going to be ugly. Partly because what measles does is it eradicates your immunological memory. So people are going to get really sick from measles. The other thing is, the older you are when you get measles, the sicker you get. And that is, again, partly because your immune system is age dependent. Most diseases you get when you're young, and then you're resistant, or your immunity is topped up, which keeps you resistant. If you get, so when a disease first establishes in a population, the average age of the population is the average age of infection. So you get many older people infected, and it looks really, really nastier. As it moves to younger people, it appears to be less virulent, but it's mainly because younger people have stronger immune systems. So that's one aspect. That's when it becomes endemic. So going back to your other questions, is some earlier work that I did and other people have done in different forms. Humans have had over 500 different viral and bacterial types of pathogens. Coexistence of all these things is, in a simple sense, just mediated because spatial heterogeneity. Most areas tend to have outside infection where everybody or a large number of people in one area has one particular type of pathogen, but they have a different type of pathogen somewhere else. So that heterogeneity always allows coexistence. Some sort of simple models I did back in the early 90s. If you look at the parasitic helminths, which are sort of more amenable to study from heterogeneity, because you can look at the individual parasites in each individual host. So what you find there is that a few hosts have tons and tons of parasites of the same species. Most individuals have very few. So that means that all of the a high proportion of the parasite population is in a small proportion of the host population, and for every parasite, the host has the high proportion of parasites. So that heterogeneity across the host population allows almost as many parasite species as individuals. So essentially, we're not hugely limited. Similarly, that heterogeneity means that you might have two pathogens circulating in a host population, but a tiny proportion of hosts have both of them. So relative to the effect of one pathogen on another pathogen is more manifested by what each pathogen does to the total host abundance than what it does in those co-infected hosts. Because reducing host abundance reduces transmission efficiency. Species A that puts the host population down reduces the ability of species B to establish. The actual interaction between A and B in a co-infected host is a tiny proportion of each parasite's population.
Thank you very much, Andy. I have a question here. There is a hand by Simon Castillo. Go ahead, Simon.
All right. Thank you, Andy, for your talk. Very inspiring for these times. So I had a question that you already partially answered, related to how the age structure of a given population may shape kind of the virulence of a given pathogen, for example. So that is tightly related to the organism's age. So my question is basically, we can expect like a linear relationship between age and virulence, or something more like non-linear, such as, for example, that we see in cancer incidence that is quite sharp after 50s. Do we see the same in like pathogens?
I mean, it plainly varies between different types of pathogens, and we, data on this is relatively poor. The classic study is by a guy called Panem, who looked at measles in the Faroe Islands, and also there's wonderful studies by Andy Cliff on measles in Iceland. But there, to understand, to be able to quantify that effect, you effectively have to have a population that's never been exposed before, and then see what's the relative mortality rate of people of different ages that get infected. So what Panem could show in his studies, that measles hadn't been present in the Faroe Islands, which are an isolated group of islands halfway between Britain and Iceland. They'd had an outbreak of measles and then no measles for 70 years, and then it got back in. So effectively, anybody who was over the age of 70 was fine. They didn't get sick. But everybody who'd been born in the last 70 years, the younger people didn't die very rapidly, but the older people died much more rapidly. And there was a beautiful exponential curve that up until age 60, if you got it, your chances of dying were higher and higher and more gruesome. And then if you'd been exposed before, you were still resistant 60 years later. Now that's, again, curious, because part of the way the measles operates is it wipes out your immune memory to everything you've had before and takes over your immune system. And then you have to get sick with all those things again before. So part of that is the younger you are, you've only been sick with a handful of things before. So you get sick with them again, but you quickly build up immunity for. If you're old, you've got 60 years of diseases you've had that you're all going to be exposed to again. And that in an older person is going to be much tougher. Now you also have, again, it relates in a less extreme way to the bats having hollow bones and therefore having a different type of immune system. In other mammals, as you get older, your thymus begins to atrophy. So once you get to 60, your thymus is more or less atrophied. That's where you produce T cells. So your store of T cells that you can produce once you're over the age of 60 begins to diminish. So you're obliged to that. If we were to look at influenza, I mean, there's all this thing that because of that T cell argument, the old people are always first in line for an influenza vaccine. But influenza is constantly changing. So you're worried about people getting an influenza they haven't had before. But if you're 60 or 70, you've seen so many types of influenza that you've got massive cross immunity to anything new that comes along. And it's the young people who haven't seen influenza before who are actually more susceptible to influenza because they have no immunity for it. So part of me would say, no, don't give the old people priority for the vaccines, give the younger people priority because they need to boost up their immune systems to start having immunity to it. So again, it varies between different pathogens. And it's also lots of the pathology. I always think of it as the Tom and Jerry cartoons. The virus is the mouse that comes in and does a little bit of damage, but the cat is the immune system, and that does as much damage trying to kill the mouse, or more damage, as the mouse is actually going to do. And that, the more Andrea Graham, my colleague here, has been looking at, within host models for the dynamics of COVID, and it's the immune system, and the way the immune system is responding to this is doing huge amounts of damage. And it seems to be doing that in a very different way in old people than it is in young people. And this sort of assumption that young people aren't getting it, we're beginning to see hospital cases of kids with it that are very different and very nasty.
Thank you.
Thank you, Andy. So we have another question by Evandro. Please go ahead.
Can you hear me? I'm not sure.
Yep.
This thing.
I can hear you.
Okay, so Erl, thanks for the presentation. It was super interesting. I was wondering about something you show very early on in your presentation, this very nice cumulative curve of various pathogens. And I'm wondering whether it's something else, whether it's known a curve of, other viruses that are not necessarily pathogens to humans, and how that can be compared to the ones you show. See, I'm thinking on a spillover and other forms of phenomena that might be driving the plateau on that curve.
Yeah, no, that's one thing we're trying to get. There's groups of us working on different aspects of COVID, but one with Peter Daszak, the guy I showed the picture of with the Nipah, he has a group of people that have been going out sampling lots of bats and small mammals in different parts of the world, and something different. So again, it shows why you need to have ecologists involved in this type of problem. So what's the species accumulation curve look like for viruses as you sample more and more individual bats of the same species and then bats of different species? So can you see how many viruses there are out there? How many of them are shared between different bat species in the same area? And if those are bats that are going into the wildlife trade, how many novel species of viruses are there out there that are in the wildlife trade for those bat species that might potentially come across, and what proportion of them have previously come across that we might detect serology for in humans? That, I think the curious thing is here, although we say we detect 2 new species of virus in humans every year, we probably get challenged by 1,000, and 998 of those don't establish because we're just a useless host for them. Or maybe one of them gets into some humans with a subtly different age or prior infection profile and begins to take off in those people. And so it's trying to get at these figures of, what is the community ecology of these viruses, and how does that affect the abundance and diversity of things out there that may or may not come across.
Thank you.
Thank you, Andy. So it's really interesting because, actually, I think that around 8% of our genome comes from retroviral invasion. So we have been dealing with this forever, for millions of years. Our lineages has been kind of adapting to and also kind of taking viral genomes in. So I wonder, maybe bats have more viral genomes that we do, and because they're more resistant. But who knows at this point?
There was a nice paper about a month ago that says, actually, the diversity of viruses in bats doesn't seem to be different than the diversity of viruses in non-volant mammals.
Huh.
Certainly, more longer-lived species, yet you have a longer chance to accumulate pathogens.
Right.
So you've got to allow for the fact that the bats, by flying, live much longer. But once you rescale by all those things, there doesn't appear to be a significantly higher diversity of viruses. But this fact that they're dealing with a very different type of physiology and immune system. The things that are in bats seem to be much nastier when they can cross into humans. And this seems to be on this. It seems that the whole humoral immunological response in bats works in a very different way. Those viruses get controlled in bats, but then once they get into a non-volant species with a different immune system, can run riot. And so although there's not more of them per se, the ones that are there, once they get into humans, can be really nasty.
Right. The other effect is that maybe sometimes when humans start encroaching natural habitat and doing agriculture, moving things around, making noises, visiting caves for tourism, I know that some bats has been reported that they decrease their immune, they become immune suppressed because of stress, and they're build up kind of the viral load and start shedding like crazy. So it's like, wow.
I think this is also a big problem once they're moved into some of these markets where they're just stacked up in crates because people want to eat them live. They are hugely stressed, so their viral loads go up, and so you've got all sorts of live virus, which is at much higher levels than it would be if you were to see them in the natural habitat.
So, I have a final questions. This thing about spillover is a two-way street.
Yeah.
And as you showed that from domestic animals, it can go to wild species. And so how important is that transmission or a spillover from humans or human-related animals like pets or kind of the pigs and so forth into wildlife species? I mean, gorilla. I know that some gorillas has been reported with diseases from humans.
I mean, we're very worried that the tourists going to see the gorillas, we really want people wearing masks because we didn't want them to bring measles or influenza to the gorillas, but the last thing we want is them to bring COVID because it could wipe the gorillas out. Some people who've been collecting fecal samples from chimps and primates throughout Africa are finding drug-resistant strains of E. coli in them. And the only way that E. coli could have got that resistant would have been exposure on farms or in human populations. So certainly there's constant spread of these pathogens from domestic livestock into wildlife. And the biggest example would still be rinderpest, which caused the biggest pandemic in the world when infected cattle from Italy were introduced into Africa in the 1890s. That wiped out 90% of the game in Africa. That's the biggest pandemic we've ever seen. Yeah,
rinderpest is just a dramatic ecosystem level impact from a pathogen. It's just incredible. So we have a question.
Something going from domestic livestock into wildlife. Yeah.
So we have a question from Ek Debal from Mexico.
Hello.
Hi.
I really enjoyed your talk. I wanted to ask you about lethality because I wonder why in some countries this virus is killing a lot of people and not in others. It's just because of the morbidity factors, or do you think there is something else?
No, that's a really interesting question. I mean, I think that it's plainly the same virus. It hasn't had time to evolve to be a different virus in different places. Although we now can differentiate genetic strains, the way they're operating is very much the same. So that has to be features of one of two different things. One, it could be the age structure of the population. So if you have populations, say in Africa, which have lots and lots of young people and proportionally very few older people, it would appear less dramatic. Though our worry is we are beginning to see really nasty cases of infection in young people in the US and in European hospitals. And so those will be occurring in Africa. We may just not have detected them yet in the different parts of Africa. The second thing is that the way this pathogen seems to work seems to be massively dose dependent. The people who've been working in healthcare facilities, lots of the frontline medical staff, where they get massive exposure, have been getting really, really sick. People who may getting casual exposure or just picking it up on their hands, depending on where you first get infected, may give huge differences in the level of pathology that each individual expresses, and how that manifests itself by the way people are responding to it in different ways in different countries may well be causing those different mortalities. And those would be the first places I'd look. But I mean, we've had 4 months and we're learning huge, big new things every day. But those differences are something. It's like, well, what might be causing this? And it causes it to have different seasonal patterns in different places.
So, Andy, what about Aboriginal populations? I mean, suppose an Aboriginal group in the Amazon that is all the time really interacting.
Well, again, how that is going to depend on the age structure and how. I mean, it does seem to be less transmissible outside than inside. So if Aboriginal groups are spending much more time outside than being in houses, maybe the infection doses they get will be less, and that might in turn lead to reduced pathology.
Huh. Very interesting. Okay. Well, thank you very much again, Andy, for being with us. It's a fantastic twist from the typical SIR approach these days. So it's good to see this new kind of a perspective to understand, to better understand the COVID epidemic. So I wish you safe time in Princeton. All the best to the good friends up there.
We'll do it.
See you soon.
Yeah, and I'll send a couple of. I can send a PDF of the talk, and I can send a couple of papers that relate to the house.
Oh, that would be awesome.
That's useful. Okay.
And if you can send the PDF of your latest book, which is Unanswered Questions in Ecology by Andy, David Tillman, and Bob Holt. I happen to have a chapter there, so that would be wonderful.
Excellent chapter. It's really good. Yeah,
thank you.
All right. Good to see you all.
Thank you for inviting. Yep. Bye-bye.
I forget how it is.